Hello everyone, my name is Mason Vaugn Brown. I'm a second year biology and human physiology major, and this summer I had the privilege of working in the muscle cellular biology lab under Dr. Damien Callahan. I would like to thank the Center for Undergraduate Research Experiences for their time, effort, and patience in allowing me to do this project. So, the goal of the Muscle Cellular Biology Lab is to identify and understand the age and sex-related changes in skeletal muscle structure and function, as the two are inextricably related. We are particularly focused on how age and biological sex influence tissue mechanics, notably tissue compliance or stiffness, and active and contractile performances. To understand how age and sex influence these outcomes, we measure force, tension, velocity, power, and stiffness, all at the cellular level. We relate our results to real-life issues such as injuries in athletes, as well as preventive measures for older populations, i.e. trying to reduce falls. All of this comes from the mechanical measurements of the tissue, how much force it exerts during stretching, and how much power it generates during contraction or active shortening. By measuring these outcomes at the level of a single muscle cell, we are able to identify specific targets for potential intervention, whereas measurements of in vivo performance are often complicated by interactions of nervous, muscle, and orthopedic characteristics. However, in measuring cellular mechanics, we have also tried to understand how the connective tissue between cells impacts tissue mechanics. One of our newer ventures is that we are trying out picoseries red staining and imaging to view the extracellular matrix and see the alignment of collagen fibrils and if they have an effect on stiffness levels. So during the lab, over the summer and currently now, I assisted by preparing for experiments, restocking frequently used consumables, washing glassware, sterilizing equipment, labeling centrifuge tubes, and preparing buffer solutions to be used in a variety of our assays. Most importantly, though, I have contributed to the lab's work by translating raw data into objective outcome measures. In order to do this, we use a couple of different MATLAB codes depending on what we are analyzing, whether it be passive or active stiffness. So in this image up here, this is our biopsy room on the top. We got a laser pointer, our biopsy room up top here. And then right below here, it's hard to see, but these are our single fiber ties. This image right here is a better close-up view of these. So these are microscopic ties that we use to mount fibers to our rigs. And I'll get into more of that a little bit later. Here, I have a screenshot of one of the figures I would analyze. And in order to do that, I would click right here before this peak, and here before the peak, all the way to the very end of this figure. And this is how we get our measurements for, this is a passive stiffness analysis, and this is force versus sarcomere length. I do enjoy the data analysis I kind of put in my headphones and kind of get through it quick, feel productive, but what I really love for the muscle cellular biology lab are the biopsies we conduct. And so all our studies begin with a percutaneous needle muscle biopsy procedure, and we do this in our older, younger males and females, and we perform the procedure in a rested and fatigued vastus lateralis muscle. This muscle is located pretty much on your upper thigh. And so our primary investigator does the whole biopsy procedure himself pretty much, and we assist along with him. But we do it in a fatigued and non-fatigued state. In order to achieve that fatigued state, we have our patients do leg extensions on one leg, and we have pretty much them sitting here on our biodex, and we make them go until failure. So the muscle is truly in a fatigued state. And that's what we extract for our fatigued vastus lateralis. So our primary investigator will then conduct the biopsy, and this is where he collects muscle tissue samples, and then later on we will parse out these samples for their prospective uses. Some tissues are used for gels, which help determine if it's a type 1 or type 2 fiber, and other tissues get used in our tissue mechanical experiments. And so these experiments, sorry, this is just our biopsy, so the parsing, and then our uses. So these are our gels down here. This is where we are type 1, type 2 analysis, and this is our mechanical. We have it on our rig here, and this is view with the microscope camera, and up here is our imaging. So the experiments we conduct in lab really have to do with passive or active stiffness. The difference between the two is that the conditions we measure the force under. Active are measured while bathed in an activating solution, which is mostly calcium, and in contrast, passive is measured in an almost identical solution, except the notable difference is that much less calcium is present. The differences we look at is the ability for the tissue to stretch and how much force it generates for a given strain or fractional lengthening. Shortening is where the power is generated, and we take this measurement in relation to the cross-sectional area of the muscle. So our single fiber contractile mechanics happen on our rig located over here in our lab, and on our rig we have microscope for mounting, helping with mounting, viewing things, viewing the tissue, as well as these tiny little wells. And these wells right here are where we keep our solutions, whether that be pre-activating, activating solutions, or other baths. And then here is our length motor. There's also a force transducer on here to measure the force that the fibers are generating. And even smaller is this section right here. And this is where we mount the fibers using those single fiber ties I showed earlier, and they're placed on these hooks pretty much where the fibers are. So, like I said earlier, under our active conditions, the fiber is placed under a pre-activating solution, which will take away all the calcium from the fiber, and is then placed into a different well that will have an activating solution that saturates the fiber in a whole lot of calcium, which causes the actin thin filament to bind with myosin, which is what happens during actual fiber activation in the human body. And we measure these fiber conditions to compare force versus velocity, and the velocity is like the velocity of the shortening of the fiber. This image on the left over here is just one of our force versus time graphs for a stretch from our active clamp dataset. And so I will now analyze this figure 5 times, and to do that I have to pretty much click right here, and then here, and then here. I'll do that 5 times, and that will allow MATLAB to spit out this figure right here, as well as a table with numerical data that shows that as time increases, the force the fiber has decreases. So, saying again, the longer the fiber is allowed to shorten, the less force it is able to exert. Here, in our passive stiffness measurements, the fibers are measured in a relaxing solution. This just makes sure that the fiber is not in an activated state and that the thin filament actin is not binding with myosin. And this is replicating in the human body as well, how actin and myosin are not binding, the fibers are not activated. The fiber is then measured across different lengths, and its force is taken at those lengths. You can see in this figure from our MATLAB code that muscle tissue does not act like springs or an elastic manner, but instead the fibers have a viscoelastic property to them, where if you're holding a spring or a band, it will hold a constant force as you stretch it. However, muscle fibers will steadily relax a little bit, even though they're being held at a constant length, pretty much. This property is known as creep, when the stress or tension in the fiber is constant and the strain or the stretching of the fiber decreases after a little time has passed. So to visualize this on the chart right here, the figure, we can see how we have force as our blue line and then we have sarcomere length as the orange line. So here, you increase the sarcomere length, then you increase your force, but as you hold that sarcomere length constant, force is slowly decreasing. And this is known as the creep, where again, the stress and the tension of the fiber is being held constant. The stress and tension is the sarcomere length, but the strain or the stretching of the fiber decreases as time goes on. So see again, this is by better side. To see again, force jumps up as the stretch increases, and then force slowly returns to a lower amount. Force jumps up again as you increase your sarcomere length. Then force again creeps on down, relaxing as you're holding your sarcomere length constant. And this data allows us to build visualizations such as these. So we can see how in our OM non-fatigued, this is just our older male using the non-fatigued vastus lateralis tissue, they have a very high strain to stress. So it means pretty stiff fibers. Second stiff fibers are the younger males, non-fatigued. So we're seeing a trend here how the non-fatigued fibers are stiffer. This is kind of like in an athlete, how the muscles feel stiffer before warm-up versus after. And then we see the age comparison, how even though they are both now in a fatigue state, the older males still have a higher stress to strain than the younger males do. Finally, I would like to thank everyone who supported me this summer for my summer research experience, especially CURE, the Center for Undergraduate Research Experiences, as well as the Musculocellular Biology Lab. I'd like to thank all my graduate students and the older undergrads who helped me through the process, as well as our primary investigator, Dr. Damien Callahan. I really enjoyed conducting research over the summer, and I'm looking forward to continuing it throughout the school year in the future. Thank you.